Hi learners, good morning to you all. Now we are happy to handle in our lecture series the second topic of Unit 1 of Immunology and Immunoinformatics. The second topic is pertaining to immune cells. Okay, so like this there are immune organs, immune cells and immunological molecules. Now for the time being, in this topic 2, we cover immune cells. So the main function of immune cells is safeguard our health. It is not our intelligence, it is not our muscle, it is not our tricks that is safeguarding our health. It is our WBCs that are safeguarding our health. So this is an animated video wherein we are seeing that there is the microbes infecting our cells. Microbes are getting broken down, proteolized, and their genes are being used for the production of the peptides, and those peptides are presented through MSC molecules 1 to the outside of the cell. Okay, so like that, any infected cell will present itself that it is infected. So once it is presenting to the cytotoxic T cell releases cytotoxins. With this cytotoxins, the infected cell collapses. The infected cell dies. Not only that, the cytotoxins make perforin molecules on the cell membrane of the infected cell. So as a result of these perforin molecules, the cell is perforated. Once the cell is perforated, the cell contents are oozed out and the infected cell dies. So like that, the animated video has shown here. So like that, white blood cells safeguard our health. So the main objectives of the topic 2, that is immune cells, is to evaluate the attributes of immune cells. So we keep talking that they are the WBCs. Of course, WBCs is a very common word, but here in immunology, we have to specifically give them an attribute that they are the immune cells. And to classify the function of the immune cells, these are the two objectives with which we are now running the topic 2, learning outcomes. Once this topic is covered, then the learners are in a position to understand the types of WBCs and their attributes are known. CDs is a very common word, common acronym that is being used in all this immunological terminology, that is called cluster of differentiation. Cluster of differentiation of WBCs will be known. Then CD8 T cells role in graft rejection will be known. And anti-CD3 protects from rejection of the organs. Anti-CD3 is nothing but a monoclonal antibody. That's what in the topic 1 we're mentioning, that monoclonal antibodies are minting money. Companies are minting money through these monoclonal antibodies as they are therapeutic in value. Next. So immune cells topic is given. Just by giving the definition, closing the topic doesn't suffice. We have to give you the full explanation of the immune cells. So the definition of these immune cells are WBCs go like this. White blood corpuscles are cells of the immune system which defend the body against both infectious diseases and foreign materials. In this case, what we have to see is they are basically called white blood corpuscles rather than white blood cells. So a cell means it is static, stays at one place. Corpuscle means the one which is a moving cell. In the blood, the blood moves, along with the blood cells move. So these are the WBCs which keep moving and entrapping the pathogens wherever they come across in the lymph gland, next, lymph nodes. So these are all the various WBCs. They are basically produced in the bone marrow. So they are all basically produced from the bone marrow. So they are classified as red blood cells and white blood corpuscles and plate cells. So red blood cells do not have any function in the immune system. They are only for making the oxygen to get saturated with the hemoglobin that is unloaded in this particular RBCs. But all these varieties of the WBCs that we come across, they are all involved in the immune system, particularly the lymphocytes, monocytes, eosinophils, basophils, neutrophils. Okay. In this case, I wish to let you know two more points here. Eosinophils and basophils are causing allergy reactions. So that's what their number. Basophil is around 0.4%. Eosinophil is around 2 to 3% in number. Majority of the number goes with the lymphocytes and the leukocytes, neutrophils. So attributes of WBC, we have to see the attributes. These attributes have been collected from various sources. At one source, you may not get all these attributes. Please get to know the details of this. The WBCs are boon to our health. As I was mentioning to you, it is not the brain, it is not the muscle, it's not the liver that's keeping you healthy. It's only the WBCs. Abnormality leads to severe disorders. They protect organs. It's not only that they are protecting as a human being totally, but our internal organs are also being protected, because when we born, we have got the organ. By the time we have grown like this, so over the time there is worn out cells are there. All those worn out cells are removed by the monocytes. They protect the tissues. There is intercommunicating molecules are produced by these WBCs. They are called cytokines. Produce and mature in lymph glands. WBCs are produced and mature in the lymph glands. They get matured whenever they get activated. The activation takes place upon encountering a pathogen. And again, they are all amoeboid type. They undertake the phagocytosis. Phagocytosis is one of the main functions of this innate immunity. And they are endowed with receptors to recognize palms. This is what we handled in the unit 1, in the topic 1. That is, they are all endowed with toll-like receptors to recognize pathogens. And they are short-lived. That is another important thing, that all WBCs are not long-lived, they are short-lived. And granulocytes are involved in innate immunity. Lymphocytes like T cells and B cells are involved in adaptive immunity. All the cells are not there in both innate and adaptive. A few are exclusively earmarked for innate immunity. A few are predominantly working in adaptive immunity. And clonal selection and proliferation. These are all the attributes of WBCs. They are all enlisted by going through the total of the literature. It is available for you learners in nutshell now. So the WBC, white blood corpuscles, the name has come from the buffy coat in a thin layer of nucleated cells when they are isolated from phycol. Phycol is a density gradient centrifugation technique wherein the WBCs are being separated as a white buffy coat. The second thing is the leukocytes means white in, that is the meaning of the leuko is the white. That's what they call WBCs like this. White name has come here. So now let us see the various WBCs. The first one is the B cell. So when we come to know about only the B cell, it is almost elementary. But you should know its attributes like CD, the cluster of differentiation. When we have got the group of cells in our blood, how do we isolate only the B cell? How do we see only the B cell? That is because of the cluster of differentiation that it's having CD19 and CD20. The B, the name of the B has come from Bursa fabricius. The name also, people say that they are derived from bone marrow. Bursa fabricius is the one from that is cloaca of a bird. Cloaca of a bird, when the bird is, the cloaca is removed, the bird has become naked. Naked in the sense, it is getting exposed for the infection. So then they feel that this is the Bursa fabricius, a gland from which the B cells are being produced in the birds. But in the case of the human beings, the B cells are produced in the bone marrow. So please see here the activation. The activation term is the one which is commonly being used in immunology. All the cells are present in us, but they are not active. Active in the sense, whenever they are primed, whenever they are given a sort of a stimulus, then only they get activated. That's what now the B cell is getting activated by getting the bacteria directly at times. In most of the cases, they are being prompted by T helper cells. That's what the B cell is having. Its receptors called MSC class II receptors. Okay, so these MSC class II receptors are being presented by T helper cells. So this is the T helper cell. It is having the T cell receptor complex. This T cell receptor complex and CD4 of helper cell both anchor to the B cell MSC class II along with the piece of antigen. Along with the piece of antigen, then the B cell is getting activated. That's what the B cell activation. So once the B cell is getting activated, B cell produces immunoglobulins. B cell produces a bunch of immunoglobulins. All these immunoglobulins are produced by both plasma cells and the memory cells. But the memory cells are kept aside for the future use, and these plasma cells along with this immunoglobulins are being used for therapy. And now see, these immunoglobulins are used for these 4 types of activities called opsonization. The second one is agglutination. That is, these immunoglobulins are against the antigen. So the bacteria is having antigen. All these bacteria got agglutinated because of these immunoglobulins, and then these immunoglobulins are also useful for the neutralization. So as a result of this neutralization, this bacterium does not work further. And again, this is useful for the activation of the complement system, so that a perforation is made, membrane attack complex perforation is made, bacteria contents get oozed out. Like this, the immunoglobulins will be useful to safeguard the health of the host. So these are all the immunoglobulins. They are Y-shaped structure. They have been determined. Porter and Edelman, a Nobel Prize was given for determining this Y-shaped structure. They are about 150,000 Daltons. So then next, let us see the T cell. T cell is being identified with a marker. That marker is called CD3, CD4, CD8. So immature T cells are having CD3, and mature T helper cell is having CD4, and mature cytotoxic cell is having CD8. And the T cells travel to the thymus gland from the bone marrow. In the thymus gland, they mature. Okay, maturation is nothing but the activation. They get activated upon encountering with self-antigen presented along with the MSC molecules. So if the T cells ignores the self-antigen presented by MSC molecules, such type of the T cells get mature. Like that, T helper cells, also called CD4 T cells, they are involved in the coordination and regulation of the immunological response. So let us see this particular T helper cell. As I was mentioning to you, the T cells are master immune cells. They regulate the B cells, they regulate the cytotoxic T cells, they regulated leukocyte activated killer cells, they regulated natural killer cells, they regulate eosinophils, mast cells, stem cells, macrophages, neutrophils. All the cells in the immune system are under the control of the T helper cells. That's what the T cells are called as the master immune cells. So the T cells, then let us see in that T helper cell, T cytotoxic cell. Let us see the T cytotoxic cell. T cells are involved directly in killing certain tumor cells, virus infected cells, transplant cells, and sometimes eukaryotic parasites. And both the types of the T cells can be found throughout the body. So now the other lymphocyte, hitherto we have seen the B cell, hitherto we have seen the T cell, now the last lymphocyte is the NK cell, natural killer cell, which is having the CD marker 56. Natural killer cell do not cause lysis of the cells which are having MSC class I ligand molecule. But those of the cells where there is missing self component, missing self MSC molecules, such type of the cells are being lysed. Now, natural killer cells directly kill certain tumor cells such as melanoma cells, lymphomas, and virus infected cells. Natural killer cells are different compared to the CD8, that is cytotoxic T lymphocytes. Cytotoxic T lymphocytes kill the target cell. Once the target cell presents the peptide along with the foreign peptide along with the MSC molecule, but that's not the case with regard to the NK cells. So these are the neutrophils. The neutrophils is a polymorphic nuclei. That is 3 to 4 lobes, but it is having CD45 marker. And the neutrophils are the first immune cells to arrive at the site of infection. So the B cells, T cells, NK cells come at a later stage. To begin with, there is the neutrophils, that is the first one, like dendritic cells. they come into the site of the infection. Then the eosinophil is double shaped, their number is only less, that is around 3%, and they are having CD markers of 34 and CD11. Eosinophils cause allergies. That's what the number is kept low by the nature. And basophils number is so less, 0.4%. They have been given the markers like CD33, CD36, and they are also caused allergic reaction. That means they are having histamine. Once they rupture out, the histamine is released, and the histamine causes allergy reactions. So as a result, we used to use antihistamine tablets like this mast cells. Mast cells also mediate inflammatory response. Inflammation means redness, heat, swelling, and pain. If any one of these four present, if all the four present, then we are feeling inflammation and hypersensitivity reactions. So this is what the monocyte chasing the bacterium through PAMs, pathogen-associated molecular patterns, through pseudopodia. So the monocytes are further classified as macrophages and dendritic cells. Out of all the cells so far that we have seen, the dendritic cells are given so much prominence that they are the best antigen presenters. They are having the CD141. Now, the macrophage is also one of the antigen presenting cells. So now let us see if we are having any inflamed site. So we are getting inflammation at some places. The first type of the cell that appears at the time of inflammation is the neutrophil. That is being followed by the monocyte. Then the T lymphocyte gradually comes. So the T lymphocyte takes care of the removal of the inflammation. That's what. Ultimately, the T lymphocyte is getting educated through the monocyte and neutrophils like this. So that's what the infiltration of the cells at the time of inflammation. So these are all the various cells involved in innate and adaptive immunity. The adaptive immunity, please see the B cells, the T cells are involved in adaptive immunity. But the adaptive immunity is not independent, it's not standalone. It depends upon the innate immunity function. Once this dendritic cell presents its peptide along the MSC molecules, then the T cells, B cells are involved. That means all these cells are intricately working. So for the sake of convenience of our understanding, we made them as a few cells exclusively depending on this adaptive immunity. A few cells are exclusively functioning and getting involved in innate immunity. So with this topic 2 we are covering under this immunology and immunoinformatics. Let us see the summary of it. WBCs are lymphocytes and leukocytes. They are immune competent and immune axillary cells. WBCs are very types of the distinct functions. WBCs count determines the state of health. Okay, so WBCs secrete immunoglobulins and cytokines. WBCs converse and execute. They are communicating each other. CD4 T cells are vulnerable to HIV infection. CD8 T cells promote graft rejection. Anti-CD3 depletes T cell population and promotes graft acceptance and tolerance. So a few of the study questions out of this topic is: what are the attributes of WBCs? How do organisms protect it? What immune cells are involved in adaptive immunity? What immune cells is classified as the best antigen presenter? Enlist CDs of at least 5 WBCs. So with this, we complete the unit 1, topic 2, immune cells. Thank you, one and all. Thank you.